Good afternoon. Welcome. So I just want to let you know that we are on a little bit of a tight schedule, so at 4 we'll just have time for a few group photos, but we do need to move David on to his flight back home. But I'm so glad that you're here today. Thank you so much for coming. Welcome to our Thursday afternoon lecture series. I'm Susan Gregorick. I'm the Associate Director for Data Science here at the National Institutes of Health. My role and the role of my office, the Office of Data Science Strategy, is to catalyze and coordinate data science and related activities across our 27 institute centers and offices. I'm really thrilled to introduce today's WALS speaker, Dr. David Baker, who shares the 2024 Nobel Prize in Chemistry. David's Nobel Prize was awarded for his work on computational protein design that you're going to hear about. But before we get to David's talk, I just have a few quick announcements. The first is that if you're interested in the continuing medical education credits, the code, I don't know if it's not up there. Okay, it's 67222. So that's 67222. Okay, there it is. Good. So for those who are watching remotely, you can submit your questions to the speaker at any time during this lecture. Simply click on the button that says Send Live Feedback, which is below your video cast window. Then type in your message and we'll relay it to the speaker at the end of the lecture. So now I'm going to introduce Dr. Baker. He received his undergraduate degree from Harvard and joined UC Berkeley to study protein transport and trafficking under Randy Schekman. He then studied biophysics with David Agard at UCSF and as a postdoctoral fellow. David joined the University of Washington School of Medicine in 1993, became a Howard Hughes Medical Institute investigator in 2000. In 2009, David was elected as a fellow of the American Academies of Science and Arts. I met David in 1999 at CASP-3, the critical assessment of prediction of protein structure. He was at that time interested in the idea that there was a finite number of different local structure elements in proteins with distinct preferences for particular amino acids, and that should have, in principle, have distinct local sequence patterns, and when you would find those in multiple sequence alignment. And as a result of his knowledge-based sequence approach, he did quite well in CASP-3 and laid the foundation for significant research into computational protein structure prediction and design. You may be familiar with some of his work, Rosetta, and the distributed computing program Rosetta at Home, and then the game Folded at Home. He's gone on to do really exciting work that we're going to hear about today. What you may not know is he maintains an active experimental research lab in addition to his computational research lab. His two efforts work synergistically to understand complex biomedical and biochemical function. They develop proteins that can toggle on and off for switches that act as drug candidates. He looks at peptides that can promote targeted immune activation and generative AI to find solutions for undruggable disease targets. What's less known about David is that he is an incredibly committed mentor to his students. He's fostered probably over 100 now doctoral and postdoctoral trainees, many of who've gone on to become independent researchers. He has a number of awards apart from the Nobel Prize. In my field, he's won the Overton Prize Award in computational biology. He's currently and remains a professor of biochemistry and HHMI investigator and the director of the Institute for Protein Design at the University of Washington. The title of his talk today is Design a New Protein Functions Using Deep Learning. And with that, I am super happy to invite Dr. David Baker.
All right, thanks. All right, well, thanks, Susan, for that very nice introduction. So today, my talk has kind of two parts. In the first part, I'll tell you a little bit about how we use deep learning to design proteins and sort of give you sort of an overview of some of the sort of the variety of problems we've applied this to. And then in the second part, I'm going to focus on one particular problem, the problem of designing new enzymes, new proteases completely from scratch. And because I want there to be plenty of time for questions, I might that second part I might skip a couple slides. I didn't realize how time constrained we would be. So, but okay. So, let's see if I can do this. How does that pointer thingy work? Okay. Well, So in biology, information flows from the genes in our genome. Oh, interesting. Okay. From the genes in our genome to amino acid sequences to proteins which have biological function. So that's pretty much the problem of biology. We kind of go in the opposite direction. So we start with a desired function that doesn't exist. We build a protein on the computer that we predict should have that function, work backwards to an amino acid sequence which we predict would fold to this structure. We can use the genetic code in reverse to come up with a synthetic gene sequence that would encode the protein. And then, importantly, in the final step, we obtain a piece of synthetic DNA that encodes that brand new protein, and we put it into bacteria or yeast or human cells, and they make the protein. And we can see whether it actually has the function that we designed. We have to make a brand new gene each time because these are brand new proteins. There aren't any genes that exist already in natural organisms that encode them. What? Okay. Is that better? Okay, sorry about that. Okay, so I'm going to talk a little bit about the methods for designing new proteins. They're inspired by methods from computer vision, where you can take all the images off the internet, add different amounts of noise to them, train a network to remove the noise, and then the remarkable thing is if you just present such a network with completely random noise, you will get an image out of it, as shown on the top. We did the same thing with protein structures. We took all the structures in the PDB, noised them to different extents, and then trained a network to remove the noise. And we found that this generated new protein structures that looked like they could have been in the PDB, but they were actually completely new. So just being able to make up or generate new protein structures isn't exactly what we want to do, though. We want to generate proteins that have functions. And so to do this, we need to condition this diffusion process to like in the same way that you would condition an image generation model to, say, for example, make an image of a cat sitting on a table. So one way we can do that is, what am I doing wrong? Oh, there we go. Look at that. Okay, one way we can do that is to provide the structure of a target, like the insulin receptor here, and then this generative process will generate a structure that looks like it could bind the insulin receptor, in that it's very shape complementary. And so we've made many, many proteins in this way. In fact, we've made proteins that bind the insulin receptor, and they're quite interesting. We can make them into agonists, and since these bind to a different site on the insulin receptor than insulin does, they actually work in animals on insulin resistant versions. So there could be some therapeutic uses. So I'll just give you some examples of the kinds of some of the proteins we've designed. So the TNF receptor is really central to inflammation, and so many of the drugs that block inflammation work by blocking signaling. So here's a protein that's being diffused to bind the insulin receptor. It's quite a remarkable protein. It binds with 10 picomolar affinity. We have versions that bind to the human receptor, the mouse receptor, and the cyno receptor. So it's really nicely set for what you'd need for developing as a drug. And in animal experiments, this protein is very effective at suppressing inflammation. We can further condition this generative process to make particular types of proteins. We can specify not only the target, which in this case is the influenza virus, and a site, which is the stem region, but we can say we want it to be an antibody or an antibody-like fold. And in this case, the diffusion process generates an Ig fold, basically a nanobody. And when we solve the cryo-EM structure, the structure is very similar to the computational model. We can go on to design full-length antibodies, which have, as you know, 6 CDRs, 6 loops that contact the structure. And this now, we can solve the structure again. And if we compare the structure to what we designed, you can see in each of the CDR loops, the actual structure is very close to the predicted structure. And also in the immune system, we can design proteins that recognize peptides when they're presented on MHC. So, in our cells, when there's something, the way that your body can tell there's something foreign inside a cell is it gets presented on the MHC. And so here there's a peptide bound in the MHC cleft, and you see a diffusion trajectory generating a protein, which is basically reading out the sequence of the peptide. And this works very well. It's basically like a synthetic T-cell receptor. And so we've been able to make proteins for designs, for example, that are selective for peptides you want to recognize and do not bind to peptides you don't want to recognize. So we're very interested in both this and the antibodies for kind of therapeutic applications because antibodies are sort of the currency of the realm of the pharma industry, and peptide MHC recognition is really key to T-cell-based therapies. We've been designing small proteins to bind to G-protein coupled receptors, both antagonists as shown here and agonists as shown in this case. I think there's a lot of interesting potential because GPCRs are so central to human physiology. Now, the proteins I've shown you so far are folded proteins, but as many of you know, many proteins in nature are not folded. Instead, they are intrinsically disordered, and that's been hard for drug discovery because there's not really a defined structure to target. But we can actually turn that to our advantage by allowing that structure to diffuse at the same time that the binder is being generated. So in this video, this animation, what's in black here is the target, which has a fixed sequence, and it's folding up. And at the same time, the protein which is being designed to bind to it is folding up. And you can see that they kind of nestle closely together. So we've used this to generate binders to quite a few intrinsically disordered regions of proteins. And there are a few examples shown here. They bind with quite reasonable affinities, and the lower panels, which I won't go into detail here, just show that these interactions hold in cells. So we can bind and perturb interactions involving IDRs. Now, a particularly class of targets we're particularly interested in are targets involved in neurodegenerative disease, which often have intrinsically disordered regions. And for example, these are binders to different regions on the tau protein, and we've got quite reasonable affinity binders to a number of different regions. And you can see actually. Okay, is this better? Okay, sorry. So here, these are different pieces of the tau protein, and it's in cyan up above, and you can see that it's being bound in this case, in an extended beta strand conformation. So we can also design proteins that bind to post-translationally modified peptides, and they're isoforms of tau, which are phosphorylated, which are particularly associated with disease. And we can make designs that bind to these phosphorylated peptides and don't bind to the originally unphosphorylated peptides, which are on the right here. And we've been experimenting on how to use these tau binding proteins. One thing we found is if you simply add them to fibrils, many of them will actually drive fibril dissociation. We can also attach them to components of the ubiquitin machinery, the proteasome machinery, and in that case, when we express these in cells, basically what happens is the binder binds the tau. That's what you're looking at here.
So when we make this protein here, we completely eliminate the tau from the cell. And our colleagues are doing animal experiments now to see what the effect is on neurodegeneration in model systems. I've described many possible uses for designed proteins as therapies, but there's also, I think, very exciting opportunities for diagnostics. And we've been designing membrane proteins, membrane protein channels in particular, like the ones shown here, which have a pore inside them. And as we make the pore bigger, we get more conductance through the channel. And we can turn these into sensors by designing proteins that sense compounds of interest, as shown on the left here, and then fusing these to the top of the pore. And then what happens is, as you can see on the right, the pore is open, letting current flow through until the compound of interest comes along, and then the pore shuts, and we don't get current through. And we're now designing we can use these design methods to design a wide variety of pores with different sizes and interior linings. And we think we can achieve something like an electronic nose, because different pores will be affected by different small molecules. Now, to make this even more general and more robust, we are now going beyond using lipid membranes and instead using a more robust platform, which is silicon nitride. So it's possible to drill holes of about 20 nanometers in silicon nitride chips, and we can design proteins that have a shape and a surface composition that's well suited for inserting into these pores. And so these are now proteins that are unlike anything that evolved in nature, because nature never really tried to make proteins to interact with silicon. And these design proteins, in fact, do insert into silicon, and we get stable current. And so now we're very excited about using these not only for sensing, but also for applications like protein sequencing. So the proteins I've told you about so far have single defined states, but we're now we can now design proteins that have multiple states. And I'll first give you sort of a toy example. So here's a designed protein that in the presence of an effector changes from this straight conformation to a bent conformation. And down below you have we have sort of a triangle-shaped assembly that we made from 3 of these hinge-like assemblies. And when the effector is added, then when the bend happens, the triangle goes into a square. And that's exactly what we see experimentally here, where we have the trimer, the triangle, when the effector is added, going into the square. And down below we have another example where the effector drives a transition from a square to a pentagon. So this is allosteric, for those of you familiar with that term, because the effector is binding distant from the actual interface. Now, that's not very useful, but we can use this principle to do more useful things. We can make medicines that actually have an off switch. So here is a designed mimic of IL-2, which is a potent immunoactivator. And we've designed it to have a switch in it so that it turns off. And the way that works is in gray up above is this IL-2 mimic. In red and blue are the 2 components of the IL-2 receptor, the IL-2 beta and gamma subunits. And so what happens initially is the designed protein comes in, it brings the 2 receptors together, and you get signaling. But when the effector is added, it causes a conformational change, which then forces one of the receptor subunits, the gamma subunit, to leave. And when we test this experimentally, we can actually, with collaborators in the Pyler lab, they can actually see the receptors first coming together when the protein's added originally, and then getting driven apart when the off switch effector is added. And that shows up, that's sort of quantified down below. When the effector is added, within 20 seconds, all of the binary complexes have been broken, and this leads to a concomitant reduction in signaling. So In blue is if you don't add the effector, and in yellow is when you add the effector. So we're very excited about this concept now, and we're using it in a number of different contexts to make signaling molecules that either turn on or turn off when another molecule is added. It could be something just present in that location in the body, and that's how we can make sort of location-specific medicines, or it could be something that's responsive to an added compound like the effector here. Okay, so now I'm going to do a deeper dive, and I'm glad I have the microphone so you can hear me, on design of catalysis. So using the methods that I described, we and others have designed many, many binding proteins, and I encourage all of you who are interested to use these methods. They're really quite straightforward to use. And so now if you have any protein of interest, it's really quite straightforward to make binders. So we've been sort of moving on to problems like the conformational change problem I just talked about, and to catalysis. Can we make proteins which actually make or break chemical bonds? And I just wanted to give you an update on some exciting recent results in this area. The problem I'm going to focus on is protease design. Now, that's an important problem because, and it's different from the problems that we have previously worked on in de novo enzyme design, which have typically been on problems where there's a pretty activated substrate, a pretty easy chemical reaction to carry out. But the amide bond is very stable. And there's another problem, which is that if you're going to cleave a protein substrate, you have to correctly cleave it in the right place. And so you have to be able to bind a flexible disordered protein. And the work I'm going to tell you about was done by graduate student Ho-Jae Choi and postdoc Angie Chen, who are both absolutely terrific. Here's the, let's see, maybe I do it here. Maybe not. Okay. Well, I have to not do it, and it will do it. So an ester bond, which is the upper reaction, has a half-life in buffer of about in minutes to hours, whereas an amide bond, like in the peptide backbone, has a half-life of hundreds of years. And that's why you can take many proteins and let them sit on your bench for months in buffer, and if they don't misfold, they're probably totally fine chemically, at least as far as the backbone goes. So this is not an easy bond to break. So as sort of a lead up to this work, we had designed proteins which catalyzed that easier reaction, the ester hydrolysis. And the way we do that is we specify the active site, and then we carry out the diffusion process basically with those residues constrained. And so then the protein has to fold up into a structure which can hold those residues in the right place. And so after that, we obtain synthetic DNA, in this case encoding 96 different designs. And we were very happy, this is work by Seth Woodbury and Dong-Hyo Kim, we were very happy to find that, or they were very happy to find that a couple of these designs had quite high activity. And so this was the ester bond. I said it was easier than the amide bond. I mentioned the problem of binding a disordered protein, and as I showed you, we had been simultaneously developing methods for binding IDRs. So now we had a way of scaffolding active sites and a way of binding disordered regions. So we kind of put them together to try and tackle the protease problem. So Ho-Jae worked on chose to try to make a protein, an enzyme using a catalytic triad, which has an active catalytic cysteine that's a nucleophile. And she chose to make a metalloprotease that uses a metal ion to activate a water to cleave the peptide bond. And we developed a method called RFD2MI, which where you specify the scaffolding residues and, in this case, the peptide substrate, and it builds it builds a protein that scaffolds the active site and also interacts with the that disordered peptide. And so we get out solutions which have not only an active site but bind the substrate peptide, which is shown in gray here. So I'll tell you about the cysteine proteases first. So the way that Ho-Jae first screened these was by having an enzyme, then the substrate, and then the cleavage site was internal to the chain. And he was very excited when he tested these designs just on an SDS gel. If the cysteine isn't present, if it's mutated, we get the full-length uncleaved protein. But all of the designs were cleaved. So this wasn't true for all of them, but a significant fraction of the designs just worked right out of the computer. So then took these designs and tested them in trans to see where they could cut a peptide that wasn't on the same chain. And he had designs in his set which were quite good catalysts, and mass spectrometry confirmed that these designs were cutting at exactly the desired site, which is indicated there. And he was able to solve structures of a number of these designs, and the structures were very close to the design models, with the peptides laid out exactly where they were supposed to be, and the cleavage site residues very close to what they were supposed to be. And as I said, the things that we're generating are really new proteins. Pretty much everything I've shown you is a completely brand new protein with new folds and new sequences that not seen in nature before. So Anshi's similarly had success with her metalloproteases. So on the top is the cis case, and she can add a zinc chelator to remove the zinc, in which case no cleavage happens. That's the upper gel on the right. But if she adds zinc, she gets cleavage and release of the protease. And this also works in trans. Again, in the absence of zinc, the full-length substrate is only is observed. In the presence of zinc, it gets cleaved. And she mapped the cleavage site by mass spectrometry for many of her designs, and it was always in exactly the right place. So these enzymes are active and they're specific. To confirm that they were working through the design mechanism, a blow-up of the active site is shown in the inset there, and then below is the effect of knocking out individual residues, and you can see that the knockouts of the catalytic residues immediately around that were part of the mechanism all completely block activity. And this enzyme is quite a good enzyme. It proceeds carries out up to 200 turnovers, so it can proteolyze many different protein chains. And she was able to solve structures, and again, they had quite high accuracy. So this is exciting. So these are brand new de novo designs which can catalyze this very difficult reaction. Now, the designs, and this is sequence specificity. So because you want a protease to be sequence specific. So this is Ho-Jae's cysteine protease is on the left, and you can see it's cutting its desired target, but even fairly subtle mutations knock out that cleavage activity. And Anshi's designs were similarly specific, except she obtained one, which is a pretty broad spectrum protease. So the activity, we wanted to know why the activities weren't even higher. They weren't quite as high as the best native enzymes. And we probed this by making mutations of every residue around the site to see what increased the activity. And what Ho-Jae found was that there was, and we could see this in the crystal structure, the histine in the catalytic triad sometimes would get distracted and swing out to interact with a polar residue which had been included in the design, the serine and tyrosine 41 and 44. And on the right is just the mutational data. The black squares are the original sequence, and the red colors indicate places where mutations improve things. So, for example, that tyrosine to isoleucine improve things because isoleucine can't interact with histine. And we saw similar things at the serine, also put in positive charges, just things that wouldn't distract the histine away from the active site. And when we've developed methods for generating ensembles of structures, and when we apply those to native enzymes, the active site residues are very crisply held in place. But in some of the initial designs, that wasn't true. But when Ho-Jae combined those substitutions that were increasing pre-organization, then the site was much better defined. And now the enzyme has gotten even better than it was before, with a substantial rate enhancement over background. And Angie found similar things, that most of the positions in her design were absolutely perfect. There was no substitution which would increase the activity. But there were several positions, again, where increasing the active site pre-organization seemed it was important. So changing this phenylalanine to a tryptophan increased activity, likely by keeping the metal chelating side chains and the general base more oriented as they were supposed to be. And we were excited to see this result when she included those sort of active site pre-organizing mutations together. She got the Kcat over Km, sort of the catalytic efficiency of her design, was considerably greater than that of TEV, which is a widely used protease. So we can now make new proteases from scratch that are nearly as active as native enzymes, or more active than some. And this is a rate enhancement over 10 orders of magnitude. So what did we learn from all of this? Well, first of all, that we can now, these generative AI methods are now good enough to place atoms in space with enough precision to catalyze even quite difficult chemical reactions. And so because of this, we can now design reactions that are, design enzymes that catalyze high activation barrier reactions. We learned that we need to improve the methods to get still better active site pre-organization. And it's interesting because we didn't really explicitly consider dynamics in achieving these quite high activities. So I think they're very exciting applications. I think catalytic therapeutics could have some real benefits over current types of therapeutics, which are mainly antagonists. So if you think about an antagonistic antibody, you need to put a lot of that antibody into your body to block all those interactions. Where if your binding protein for that same target has a proteolytic site in it, it can destroy the target. And so one drug molecule could eliminate a large number of targets, say a viral surface protein or a surface protein on a pathogen. We're very excited, as I sort of indicated earlier, about using this in the context of neurodegenerative disease. And more generally, this design approach can be applied to a wide range of bond-breaking and bond-making reactions. For example, we're going back to organic chemistry textbooks and making catalysts for SN2 and SNAR reactions and ligases. So it's a really exciting time. We've developed now diffusion methods that work at the atomic level. This is called RFD3, which are even more effective at scaffolding complex sites. And she has used those to make designs that cleave TDP43, which is also involved in amyloid disease. And so we're very excited about pursuing this area of therapeutic proteases. So I've outlined some of the uses of design proteins, and there's really a very exciting, given the wide range of things that proteins do in nature, I think there's a very wide range of problems. For example, we're designing proteins to break down plastic as well currently. And I just want to thank the broader community and people in my lab, and it's really been a lot of fun, and thanks for your attention.
There are mics in the aisle, and I'm sure there's lots of questions. There are a few online, but happy to start in the room here.
Go ahead. Hi. I have two very brief questions. The first one, you mentioned a lot about enzymes. I was wondering what you think the next frontier of protein design is. Is it maybe accurate conformational prediction? Is it maybe like attacking a specific mechanism like prions? Yeah.
Yeah, so I think the current frontier of protein design is combining the modalities that I described. So I talked about binding, I talked about conformational change, I talked about catalysis. So designing machines and motors is really exciting now. And so you could imagine, so things like walkers, things like disaggregases, and you could imagine, one of the problems we have in our bodies is there's nothing in circulation that does general quality control. We have chaperones inside cells, but nothing like that outside cells. So you can imagine protein therapeutics that are basically machines that go around repairing damage that work outside the cells, where the fuel is, say, something in your diet, like a triglyceride. So that's I think, some of where the current frontier, or like artificial ribosomes that put together completely new types of compounds.
Second, very brief question. I've seen that you, in your last year of undergrad, you switched your major from philosophy to just pure biology. I was wondering if you could just speak to how important breadth or new perspectives are in even highly technical work.
Yeah, I actually think, science changes all the time, and the techniques change all the time. So I think having breadth is good. Like you can't, even if you're a world expert in something today, chances are that in 5 to 10 years, whatever you knew was going to be obsolete. So I encourage students in the audience, learn different things. I think it will stand you well later in your careers.
Thank you.
Thank you very much for your talk. My question is about when you looked at designing proteins to bind to intrinsically disordered proteins, did you see a trade-off or an increase in off-target binding? And where I'm going with this is, how do you distinguish between looking for off-target as opposed to non-specific, right? Because you're using structures that have never been found before. Even something that's specific could still have an off-target effect if it shares a similarity with another structure, right? If it's in a similar area of sequence space.
Yeah, well, that's a good question. So we've been collaborating with Matthias Mann's group, who does a lot of mass spec proteomics. So we've sent his group about 150 different designs, both to folded proteins and to disordered proteins, and they have just taken, full cell lysates and seen what sticks to our design, and they can read that out by mass spec. So some of the designs are absolutely specific. They only bind their target. Other designs bind the target, and then they bind other targets as well. So an example of an intrinsically disordered protein binder that we made was to MYC, because we were interested in anti-cancer things. And that design is very interesting. It really shuts down MYC function and shuts down the growth of cells that require it. That design was quite specific, but we do have other IDR binders which are less specific. So we're still early in the early days of determining. And then as far as distinguishing, I don't think any of them are truly non-specific. I think it's what you said, that some of them have specific off-targets, and we need to look more at these results to understand, when that happens.
Makes sense. Thank you.
Yeah, Dr. Baker, I just want to say what an exciting talk this was. And my question might be a super basic question, but I'm wondering if you take binding kinetics into account, competitive binding with your design protein with other proteins that already exist in vivo system, and if your design protein compete with them for binding, or if other proteins compete with your proteins for binding.
Yeah, that's a really important question. So if you're making an antagonist, something that should block an interaction, then you need to outcompete the thing you're competing. So that's just a matter of affinities. So we need to make designs which are higher in affinity than the interaction we're trying to block. And so the typical affinities we get out, I mentioned those TNF receptor binders were in the single-digit picomolar range. So they do outcompete TNF, binding the TNF receptor. Whenever you're trying to manipulate a protein-protein interaction, you always have to pay attention to the concentration and the affinity of your design compared to the natural one. But we've made antagonists for a pretty large number of cytokine receptors, for Leonard's Warren's here, so that's so what we've been able to do is, this is a little bit of an aside, but we can take pairs, proteins that bind to cell surface receptors, the designs, and then link them together, and those designs will bring the corresponding receptors together, and we get signaling out. So it's not quite your question, but we can make these binders into agonists by fusing them together, so we bring together receptors, and then that leads to their intracellular kinase domains cross-phosphorylating. So anyway, there's a lot of ways to get biological activity out of these designs.
Thank you.
Hi. Earlier in your talk, you mentioned designing proteins with conformational changes, which are more like hinge motions.
Yeah.
So I was wondering if you have any plans for like larger changes, such as fold switching or metamorphic, or just more broadly, what you think like the importance of fold switching proteins?
Yeah. Let's see. So the fold switching I think that will happen to the more disordered proteins that we bind, where they'll really change conformation dramatically when we make binders to them. One type of motion I didn't describe, we can take the design catalysts I described and use the diffusion to have them populate another conformation, and then be able to drive them by addition of effector into the catalytically active state. So there we can make allosteric enzymes, but we haven't designed things which are undergoing a full fold switch.
Thank you.
Hi, David. Glad to see you again here, and so many progress since you came last time. We know that although AlphaFold shared the Nobel Prize with you, but from CASP16 in 2024, we know that there are a lot of problems, a lot of things AlphaFold hasn't been able to solve. For example, nucleic acid structures or complex, or like intrinsic disordered regions and antigen antibody complex, things like that. So it's amazing that you can design the protein to bind intrinsic disordered regions. So 2 questions. So I'm curious how the detail, how even though the structure is unknown, but your design can bind it. And then the second question, I noticed that in your design, it doesn't seem to have binding nucleic acids. So what's your take?
Right. So those are 2 very important questions. The first question is, the way that we can bind intrinsically disordered regions is we sort of take advantage of the fact that they're intrinsically disordered. So they sample different possible conformations, and they adopt a structure which can be readily bound by the design. So let's say you have a disordered region with 2 hydrophobic residues. Those are easy for a protein to bind to. So it'll adopt a conformation where those 2 hydrophobic residues are sticking straight into the binder. And so it's basically the concept of induced fit. We don't know what the structure of the disordered protein is in isolation, but we induce it into a structure. And then as far as nucleic acid goes, we have some exciting recent results on being able to design sequence-specific DNA binding proteins. So I will say that we have been able to design proteins that bind DNA for a couple of years now, but they didn't bind with high sequence specificity. And so now it looks like we can finally design proteins with high sequence specificity, but we need to do more, and maybe next time I come, I'll talk about that problem.
All right. We're looking forward to it.
Okay, thanks.
Hi. I'm wondering if you could clarify how you produce these de novo designed enzymes. Do they fold well in living cells? Do you make them in inclusion bodies, in vitro?
Yeah, that's a good question. Yeah, it's one of the things that we found when we first started designing proteins, which was a very long time ago, is that design proteins were just better than naturally occurring proteins. They folded faster, they they were more stable, they expressed at higher levels. And that's true for the way that we're making these proteins. Almost all the ones I showed here is we just use standard expression vectors, PET vectors for E. coli. So and we induce expression for an hour, and then we break the cells open, and they have just a nickel affinity tag, and so they're almost always soluble and highly expressed. It turns out that, and actually for people who've worked on design proteins, which is students in my lab, when they have to go and purify a naturally occurring protein, they complain bitterly because they're so difficult to work with. I think the reason for that is that naturally occurring proteins are constantly getting hit by mutation, and so they only have to be good enough. Whereas the design proteins, the design method is putting in everything it knows, so they tend to be more perfect in some way.
Okay, thanks.
I just want to remind you that lactose synthase was the very first example where the flexible loops were shown the role. I hope you know that. It was the very first example.
Yeah.
And that interacts with the alpha-lactalbumin, the flexible loop of glycosyltransferase, and then changes the activity of glycosyltransferase. So that example is exactly what you are predicting, how it is basically affected. Both the acceptor site is changed, that is from glucanol to glucose. That's how lactose is made. But that interaction is so important that how mammals have become what we are today because of lactose synthase. And that example is exactly what you have basically described. That was done.
Thanks.
Okay. So I have
well, thanks, everybody.
I have a couple questions online.
Sure.
Still holding? Still going strong? All right. Okay. So some hard-hitting ones here. How do these de novo protein structures compare in stability to biological proteins? He's essentially asking melting temperatures. How do they compare? Do the synthesized proteins have the same stability in the hydrophobic core?
Yeah. Right. So, the design proteins are typically quite stable. Most don't melt until just start melting at like 95 degrees C's. And but there is an important caveat, which is this was a pretty misleading talk because I just showed you the designs that work, and there's an awful lot of them that they don't express. There are some that don't express at all. There are ones that just are gook at the bottom of the tube, or there are ones that express well but don't catalyze the chemical reaction. So those ones, it's not that we don't love them; they just don't make it into the talk, but they're there. So the success rates really vary from problem to problem, but when they work, they generally are quite stable. And the reason that they have exactly what you'd expect. They have perfectly hydrophobic cores. They have charged exteriors, and so they obey the rules that that structural biologists are familiar with. They generally obey those rules better than natural proteins do because these deep learning methods have just really learned patterns off from the protein structure database.
Okay. One more from please.
Thank you very much. First of all, I'm curious whether you also work on antimicrobial reagents.
Yeah, so we've
and
been
in this case, you probably can organize the system to minimize the risk of escaping from your reagent by minimizing.
Right. The challenge has been for attacking microbes. Is that they have the outer cell membrane. So we've been focusing on the adhesins and conjugate. Basically, almost all bacteria have things that protrude beyond the peptidoglycan, so they can contact the target. So we've made proteins which bind those. For example, in Klebsiella to FIMH, and those are pretty effective. I'll tell you the biggest problem now, and we've made things that neutralize many viruses. I'd say the biggest problem right now is we can design binders that are very high affinity, and that we can write a paper on that. But trying to get them further into the world is really hard. If we design something that's for oncology, then we can start a company or someone will want to license it. But for infectious disease, it's a sociology problem. It's just who's going to pick it up, right? I mean, so it's a lot more than designing the binder to get it out into the world as a drug. Yeah.
Thank you.
Maybe one more from here. You go. And then I'll have one line, and we'll call it a day after that.
I was also wondering about designing IDPs that remain disordered upon binding.
Yeah, that's hard. I mean, I don't think there's many examples of that in nature where you have two IDPs. Well, you could have IDPs well, hold on. Many of our binders, if you look at the structure of the IDP, it is not ordered. It still looks scraggly when it's bound. I showed some examples where it forms a beta strand, but there are other times when it forms something scraggly. What we haven't done, and I'm not sure there are examples of in nature, is having two IDRs bind each other without adopting some kind of folded structure. But yeah, if you look through, we published a couple of papers on IDR binders, you'll see that the IDR regions are not terribly ordered still.
Thank you.
So maybe one more question from online. Fascinating talk. Are these unique protein therapeutics uniquely poised for anti-drug antibody responses and therefore limiting their clinical utility?
Right, that's a really good question. So these are brand new proteins. They could evoke an immune response. What I can say is we've had a couple of these proteins in human clinical trials, and they have not elicited strong responses. You might find that surprising, but they're really stable and small, so they probably don't get, peptides from them probably don't get presented that well on MHC. But there is a flip side to the coin, which is if you compare, well, a good example is interferon gamma, where I just heard a talk the other day which suggested that a lot of the really bad responses to viruses and viral vectors have been due to autoantibodies against interferon gamma. Now, if you have a medicine that's going into people which is a synthetic protein that has the same effect, you may get autoantibodies, but those antibodies against your drug are not going to cross-react with the native cytokine or so you don't have the risk of disease due to recognizing a host protein. So I think the immunity question kind of goes both ways. It's you may be slightly higher chance of getting anti-drug antibodies, but you don't run the risk of those anti-drug antibodies then causing disease.
Thank you. Oh, okay. You're so cute.
How did you get the idea to do this?
Awesome question.
Yeah, well, that's a good question. I mean, I didn't really have the idea to do this, until it kind of happened. I mean, I was just kind of like trying different things out, and actually, I didn't really do any of this myself. It was my students who did this. So, and I think the ideas come from all over the place. They came from people talking to each other, and from people thinking about what might be interesting or useful to do, and what they want to solve. Like, do you want to cure disease, or do you want to break down plastic? So it's a very good question. I think the ideas kind of come from all over the place.
Thank you.
Thank you, and thank you for joining us today. You're all invited to a very modest reception around the corner in the library, which is over here. So please join us. We will be happy to welcome you. And thank you, David.